Nanidah Binti Jusu with my matrix number 2021486734 from UiTM Kuala Pilah, Binti, Negeri Sembilan. I'm from AS1141B2 and my group name is mica.
Hi guys, Assalamualaikum warahmatullahi wabarakatuh. My name is Nureen Iwana Qistini Binti Mohd Zairee. I'm from AS1141B2 and my student number is 2021650472.
Hi, I am Faqihah Binti Suhadi and currently I am studying microbiology in UiTM Kuala Pilah. My group is AS1141B2 and my student ID is 2021461156 and my group names is mica.
You guys must have been wondering, what is binary fission? Binary fission is a sexual reproduction that separates the cells' bodies into two new bodies. Binary fission only happens in prokaryotic cells, such as bacteria. In binary fission, it has potential to grow to the size of the original. In binary fission, cells that are divided are identical to the parents. It means how the parent looks like, the daughter cell will look just like the parent. However, binary fission do requires a right condition to continue reproduce. Binary fissions need a warm environment and a plenty nutrients. It is often classified into transverse, longitudinal, and oblique types based on the axis of the cell separation. These are the example how binary fissions works. In first diagram, we have the parent cell. In second diagram is when DNA duplicates. Next is when the cytoplasm divides. And the last diagram is when the two daughter cells are completely produced. As you can see, the parent cell and the daughter cells are identical. Binary fission are not the same as cell cycle. Remember, they both produce new cells, but they are not the same. Cell cycle are only for eukaryotic cells. Now, let's take a look how the binary fission can be seen through. Just like cell cycle, binary fission also have phases. Binary fission have 4 phases. In the first phase, we have is replication of DNA. The second phase is growth of cell. The third phase is segregation of DNA. And the last phase is splitting of cells. As we can see, binary fission are less complicated than cell cycle. These are the benefits of binary fission. In binary fission, only single parent needed. It means it doesn't need a partner to reproduce a new cells. Second, all the prokaryotic cell can divide rapidly. It is more fast as we think. The third one is, as we know, the daughter cell are identical to the parent cell. Can you see it? Both of them are alike with each other. The last one is a parent cell can incredibly produce many cells rapidly. Amazing, right? Do you guys know both mitosis and binary fission are similar because they both are involved in the division of cell. But in mitosis, cell division helps only growth of the organism or replacement of cell. Binary fission is not only the growth of organism, but it is a process where the organism reproduce and produce more bacteria. Now, let's take a look to the similarities of the binary fission and mitosis. For the similarities, firstly, both binary fission and mitosis produce two cells with identical genome. Secondly, binary fission and mitosis are used by eukaryotes and prokaryote cells. Thirdly, both are provide for genetic diversity. For the differences, firstly, the function of binary fission is for reproduction. The function of mitosis is for cell growth in multicellular organisms. Binary fission occurs in prokaryotes and eukaryote cells, but mitosis only occurs in eukaryote cells. The stages of the binary fission are replication of DNA, growth of a cell, segregation of DNA, and splitting of cell. The stages of the mitosis are prophase, metaphase, anaphase, and telophase. Dividing bacterium, like a human cell, needs to copy its DNA. Unlike human cells, which have several linear rod-like chromosomes encased in a membrane-bound nucleus, bacteria have only one circular chromosome and no nucleus. The bacterial chromosome, on the other hand, is found in the nucleus, a specialized area of the cell. There are several steps involved in the binary fission process of prokaryotes. As what we know, prokaryotes possess a single circular of DNA strand. The first step is replication of DNA. It involves the DNA strand being replicated in response to a replication signal. The bacterium uncoils and replicates its chromosomes, essentially doubling its contents. The second step is growth of a cell. As replication continues, the two origins move towards opposite end of the cell, pulling the rest of the chromosome along with them. After copying the chromosome, the bacterium starts to grow larger in preparation of binary fission. The cell grows and elongates by creating more cell membrane and cell wall. It is followed by an increase in cytoplasmic content. Another prominent trait of this stage is that the two strands migrate to opposite poles of the cell.
Step 3 in binary fission is segregation of DNA. The segregation of DNA in bacterial cells is an efficient process that assures every daughter cell receives a copy of genomic and plasmid DNA. Unlike in the higher cells, the segregation of the bacterial chromosome is a continuous process in which chromosomes are separated as they are replicated. During this phase, the cell will elongate with a septum forming at the middle, which we will see further details later. The chromosome will move away from each other while the FtsZ protein moves to the middle. The septum will form along the line up FtsZ protein at the end of the process, creating a partition and acting as a divider for cytokinesis to occur. Oh, by the way, septum is a dividing wall or membrane, especially between bodily spaces or masses of soft tissue. Now you may be wondering, what is FtsZ protein? FtsZ stands for filamenting temperature sensitive mutant Z. FtsZ is a protein that will assemble at the future site of bacterial cell division. While eukaryotes have the protein tubulin that help in mitosis, prokaryotes have FtsZ protein to assist in binary fission. Now let's look in details the process of segregation of DNA. First will happen cell growth and separation of chromosome to the opposite poles. As said before, segregation of DNA is a continuous process. So, as the cell elongate, the chromosome replicated and at the same time separated. At that moment, FtsZ protein in the cell will also move in and gathered in the middle of cell, slowly forming FtsZ ring. Well, it didn't stop there. FtsZ protein moves further in, lining up in the middle of the cell, which will become a sort of guide where septum will form. As septum is formed, the cell is ready for the next step. The last step in binary fission is cell division. In this phase, cytokinesis will occur. Cytokinesis is the process by which the cytoplasm of the parent cell divides into two daughter cells. Material of cell wall and plasma membrane will accumulate at the septum, forming new cell wall and plasma membrane along the septum. The cell then pinches into two, separating and forming two new daughter cells. The FtsZ protein then disperses throughout the cytoplasm of both cells. Just like this, the cycle repeats on and on. By the way, fun fact, organelles such as mitochondria also use this means of cell division to increase in number. In conclusion, binary fission occurs in prokaryotes as means to reproduce and increase in number. It involves 4 steps, which is replication of DNA, cell growth, segregation of DNA, and cell division.